printing process, how to print properties on the surface. Then we dig into the area and the environment around the cells and how to optimize it. So today I won't discuss anything about printing actually. I will show you our results related to hydrogel development and also how to tune the release of growth factors. So it's really the microenvironment around the cells that we want to optimize. And so what is important when we create the environment? We think that we want to mimic the viscoelasticity around the cells that the extracellular matrix provide in a natural environment. So when we develop our hydrogels, where we see the cells, we also want to include the viscoelasticity. We also want the hydrogels to remodel over time. So when the cells develop the tissue, the material should not only degrade, should also be able to be dynamic and remodel and adapt to where the cells want to be. That is important for us, the viscoelasticity and the remodeling properties. And we also want to know exactly when and where we make the hydrogel active and when to release the growth factors. And that we will do with light. And altogether, both these, that we exactly know when and where we release growth factors and that we have viscoelasticity and remodeling, we use shift-based chemistry. So what I will provide you today is only, the results will only be related to our shift-based chemistry and actually dynamic covalent chemistry. And what that means is that we work with amines, hydrazine and oxyamine. And when you react those with an aldehyde, there is an equilibrium. So if you have an amine and react it with an aldehyde, you create an imine. But this imine is not stable. It's actually rather be as an amine. So in the equilibrium, you're more towards the amine. If you instead use the hydrazide and react with an aldehyde, you create an hydrazone. And then this equilibrium is not directed to any side. It goes back and forth. It can be both the hydrazide and the hydrazone. Instead, if you're using an oxyamine and react it with an aldehyde to obtain an oxime, this oxime is very stable. Then it's preferred to be an oxime and not the oxyamine. And I will be referring these to TAB, ADH, and PDO, so you know. But the important here is to know that there is different equilibriums here. So if we react in the oxyamine with the aldehyde to obtain the oxime is a stable part. So then you have, this is cross-links, right, in the hydrogel. So if you have this reaction, then you will have cross-links, the oxime. Instead, if you take the other one with the amine and cross-link it with the aldehyde, then the imine won't be there for so much. So then you won't have so many cross-links. So the properties of your hydrogel will be dependent on this equilibrium and which cross-linker you actually use. So that is the basics for the whole lecture, that we will use these cross-links in order to determine the properties of the hydrogel. And to start with, I will we have used this chemistry to release the growth factors. So I start there and then we take the hydrogel in the next part. And this work was done by Tara and Tove, two of my PhD students. Tove is now a postdoc at the Karolinska Institute, and Tara will defend his thesis in a month, the 14th of November. So the idea here was that we have a hydrogel. Actually, it will be based on hyaluronic acid. And then we functionalize it with a coumarin entity that we can irradiate so that the coumarin part releases. And then we create an active part, this yellow dot. So what happens here is that when we irradiate, from start I should also say that the coumarin-based functionality is fluorescent. So we can detect with a plate reader, for example, and confocal where exactly we have this functionality. Then we irradiate, and the coumarin part is released. We have both used near-infrared light and UV to do this photolysis. Then you obtain some active part here, some active functionality that is not fluorescent. So we can actually detect how much of this coumarin that we releases. And then our idea is that we have the growth factor with an aldehyde that we can attach to this functionality, creating this part where we have the growth factor covalently bond to the hydrogen. And then since we have a bond here that can be broken by hydrolysis, then and that they are also different, have different stability, then it would release at different time points using hydrolysis. So that is the basic part. So just let me go through it in more detail now. So what we first did, these photo cages, the coumarin-based functionalities, they are not available, so we needed to synthesize them. So we synthesized 3 of them. I won't go into detail into the synthesis. If you're interested in that, we can discuss that separately, or you can read it in the published paper. But so we have 3 different photo cages, and here you can see that this red one, if you look closely, it is an amine-based cage coumarin, and then you have a hydrazine cage coumarin, and you have a hydroxyamine cage coumarin, so that we can create these different equilibriums. So if this one then reacts with an aldehyde, we create the imine. So we synthesized these photo cages, and what we did first, before we attach them to the hydrogen, was just to see if we could break the bond, if we can release the coumarin part. So we just irradiate it with UV at different times. Here it's from 0 to 100 seconds. And this is then how the fluorescent, how it decreases. And you can see that if we have the amine cage coumarin irradiate, the fluorescence is decreasing, as well as for the hydroxyamine cage coumarin and the hydrazine cage coumarin. So by just irradiating onto these, we can see that we can release the coumarin part. So it's actually here where we break the bond, and this coumarin part is released, and then we create an active group out of this. So we could see that we had a quite fast photodissociation, which is good. And the next thing was then to attach these photo cages onto our hyaluronic acid. So we needed first to functionalize the hyaluronic acid. We also cross-linked the hyaluronic acid afterwards, and we attached the photo cages. So what we end up with is a hyaluronic acid where we have functionalities that we can use for cross-linking, and we have this functionality that we can, the coumarin cages, so that we can activate it. And we then, of course, then needed to check, so if we then attach the coumarin cages to the hyaluronic acid, is it still active? Can we still release the coumarin part? So we tried that as well. And here is the reduction of fluorescence shown, so then it increases. By time, if you irradiate, the release is increasing since the reduction of fluorescence is increasing. And that is true for all the coumarin cages. So this is actually what's then happening. So here we have, on the black dot here, you have it attached to the hyaluronic acid. And then you create, when you irradiate, you create this amine, hydrazine, and hydroxyamine group. But what we then wanted to see if this group is active. If you have an amine, can you then can you attach something? If you have it in the hydrogel, can you actually attach something to it? So we used a dye where you have aldehyde group, add it to the hyaluronic acid to just see if it reacts with this functionality. And And this dye is fluorescent, and we could see that if we add the dye, you get fluorescence to it. So we could verify that this is active on the hydrogel, and when we attach an aldehyde or add an aldehyde, it is actually covalently bonded to this active group. So this was good. We also, until now I just shown you what it looks like when you use UV. We could also show that we could use a near infrared light and a 2-photon laser to do the same thing, to do the cleavage of the coumarin-based end group. And it's, again, we could verify that the fluorescence is decreasing when we increase the irradiation time. The good thing with when you use this 2-photon laser is that you can be very specific of where you irradiate. And then it's where I come to the fact that we don't want to exactly decide where and when to activate. If we can just irradiate on a specific spot and activate that part, then in the next step we can actually add the growth factor to that exactly spot. Right? So we tested that. It's a quite small picture perhaps, but what we did was that we irradiated on a region of interest, and we could see that this region of interest, the fluorescence was less. We could also see that if we added a dye, this dye was attached exactly onto where we have irradiated. And using this, we actually, when you use a confocal, you can also go into different layers in a Z direction, right? I'm sorry, I should have made these a bit larger, but I can tell you what it looks like and what actually I want you to see. So what we did now was that we irradiated in the Z direction, going from, we didn't irradiate it on the top, but we irradiated, so this is actually a star that we irradiated between 11 and 20 micrometer down. And then at between 41 and 50 micron, we made another figure, and at 71 to 80 micrometer down, we irradiated again. And in between those, you can see that it's not irradiated. The fluorescence is the same. But where we have irradiated, we can see that fluorescence is gone. So not only on the surface, you can also go down and irradiate specific spots and make then the hydrogen active where we have irradiated. So we were quite happy with that. We have a system where we can exactly decide where to activate the hydrogel. But the next step is then to add a protein to that and make it useful. So that we tried. We added dye also here to and activate it. And we could see that by time, if we when we add the dye, the fluorescence is increasing, also in the hydrogel. But this was just like a trial to see if we can add something if the active group is active also in the hydrogel. What is more interesting is to add a protein, attach an aldehyde group, and attach that to the hydrogel, and then leave it for some time to see if it releases, since this bond will be hydrolyzed at different time points. So we use two different proteins. This is a bovine serum albumin that we first functionalize by adding this aldehyde group so they can be attached to these active parts of the hydrogel. And then we also added the dye so we can locate where it is so it becomes fluorescent. So then we have aldehydes group, and we can also detect where it is. And just to remind you now that to go through the step again so you can follow me. So we attached this coumarin-based photo cage to the hydrogel. The black dot here is the hydrogel. You have the active part and you have the coumarin. We irradiate so that you release the coumarin part, and you have this active group. Then to this active group, you attach now the functionalized growth factor or protein. So you have that in the end. Then after this reaction, you then have this imine, the hydrosign or the oxime. And then by time, since this group, as I said at the beginning, is related to the equilibrium, it's also different, influenced by the hydrolysis, at different time point, this growth factor will be released. And that we can see that the protein is released at different time points. The imine, that is the one that was equilibrium towards the amine, is released fastest. In comparison to the oxime, that is more stable, it takes a longer time for it to release. So we could verify that our ideas about this equilibrium thing and how it influenced the release of the growth factors actually is true. And we also tried that. So this was with the BCA, which is quite a large protein. We also tried it with a smaller protein, the omomic, if you know about that one. And it was the same thing. We could verify that we could functionalize it, attach it, and that it was released at different time points, just related to this equilibrium. And then I won't go into the biology part of this, but in a collaboration with Karolinska Institutet, we gave them these hydrogels, and they evaluated this release of the proteins to the neuroblastoma cell lines and how that influenced them in their growth. So in the paper that is also published, and they were really happy to see that this was possible. And the results were good. I think I stopped there with the results of the biology. I think there might be some of you that are interested in biology here, but we can discuss that also more separately or look into the paper. The most important for me and for you is to remember now that we use this chemistry to functionalize the hydrogels, and at specific spots we could decide where to activate it and that the growth factors were released. And the other thing that I mentioned I want to tell you about was this dynamic functionality and the remodeling and viscoelasticity, which is important when we design the hydrogels for our cells. And why that is important to think of is that, I mean, if you look into the native tissue and the dynamics there, you have many biophysical cues. You also have biochemical cues, and all of this together will be important for directing cell differentiation. And also that we need to have in mind that the elasticity of our different organs and tissues are very different. So we need to be able to vary the viscoelasticity. Where you have the brain on one inside, and then you have fat and muscle and cartilage, etc. So when we develop our hydrogels, you also need to tune this elasticity related to which tissue you are aiming for. And not only the elasticity, also the stress relaxation varies depending on which tissue you're using. Now I understand that it's difficult for you to see here, but so here is just an example of stress relaxation of different tissues, from adipose, liver, brain, up to heart tissue. So that's the background why it's important to tune this. And when we look into the chemistry and the crosslinks we play around with, I mean, you have the permanent covalent bonding, where you have the viscoelasticity on one end side. If you have just physical cross-linking, so non-covalent bonding, you are on the other side, right? But what we are trying to do is to use dynamic covalent bonding, and then you have much wider area to play around with related to the viscoelasticity. So that's why we're using dynamic covalent bonding, and in the literature there are an enormous amount of results related to this chemistry. And going down to look into the chemistry, it's super small. There are different kind of chemistry that you can use, where shift bases, where we're using is one example. But there are other chemistries that you can use for creating this dynamic covalent bonding. So what we did using shift bases and dynamic covalent chemistry, or not me exactly, it was actually Taha, and we had a master student, Jan Ru, that helped us, and we had Karina, she was an exchange student from Switzerland, that worked with us for a few months and helped us to do this work. And this is also published if you want to look into the details. But what they did was that they looked into, they functionalized the hyaluronic acid to create these aldehydes, and then they used these different cross-linkers, the DAB, the ADH, and the PDO, and created a cross-link structure, and they looked into how that influenced the properties, if we could tune the properties of the hydrogels. So they looked into the different shift-based bonding, just based on the cross-linkers. They varied the polymer concentration, the degree of substitution, which means how much functionalization we have in the aldehyde. And they also looked into cross-linking densities, so the ratio between the aldehyde group on the hyaluronic acid and the amount of cross-linker. And they then looked into the mechanical properties. And for doing this, we're using rheology. And here is some results of what it looked like, where you have different functionalities. So here we have hyaluronic acid, where we 13% aldehyde functionalization, and on this side you have 50% of aldehyde functionalization. And then you have the different cross-linkers here, the DAB, the ADH, and the PDO. And you can see, if we just take out one example, that if you have the cross-linker where it's towards the oxime, where it likes to be cross-linked, then you have you have higher mechanical properties in compared to if you have the imine that rather be as an amine and not cross-linked. So this was our first proof that we could vary the mechanical properties by just changing the cross-linker and taking advantage of this equilibrium between reactant and cross-linked product. We also tried if these hydrogels, then if we can use this equilibrium to create a self-healing hydrogel, and that we could. In this picture you can see the hyaluronic acid that is functionalized to 50% and 13%, and that we have used on one end side we have used the ADH as crosslinker, and the other side we have used PDO. So we cut pieces and then we put them together with different crosslinkers. So on one end side you have the ADH crosslinker, and on the other side you have the PDO as a crosslinker to see if it could heal. And we saw that after 50 minutes it was had self-healed. There was just one hydrogelatin. You could not separate it. So they healed even though they had different crosslinkers. We could also see that we then could tune the properties in both cases, that if when we combined two halves, put them together, the mechanical properties were in between the PDO and the ADH, independent on if you had a 50% functionalization or a 30%. So the self-healed were obtained. So in that sense, you could also tune it. You could synthesize two hydrogels and cut them and put them together, and you get another mechanical properties, which we thought was a smart way of doing it. We also evaluated the cell material interactions here, and we could first see that when we used the ADH and PDO, the cells really liked the surface in comparison to the TCP. We couldn't see any any toxicity at all. And perhaps more interesting is that when we cut them into pieces, we could also see that the cells they kind of chose one part. If we see that the cells on this scaffold where you have 13% functionality and 50% functionality, they have both the ADH and crosslinker. They choose to be on the side where you have 50% functionality, which we think is related to the mechanical properties there. And the same thing if we have different crosslinkers, the PDO and the ADH, it also turn out on one of the sides. It's also a way to direct the cells to change the mechanical properties and direct the cells. So we were happy with this. You could see that we could tune the mechanical properties of the hydrogens by just working with the equilibrium of these crosslinkers. The problem with these system is that they are not so stable in vitro. We wanted to increase the time where the hydrogen is stable during in vitro tests. So our next aim was then to prolong the in vitro stability, but we still wanted to keep the self-healing and injectability, and also the viscoelasticity. Otherwise, I mean, you could of course just use covalent crosslinks in order to increase the in vitro stability, but we wanted to also keep the stress relaxation and viscoelasticity properties here. So what we then played around with was another system. We used the same crosslinkers as we had discussed throughout the lecture, but in this case we compared where we have the ADH and the PDO, and just as crosslinkers as we have done until now, and on the other side we attached covalently the ADH to one side. So you keep, this is covalent bond, and then you have a dynamic covalent bond on the other side. And our idea is if you have such a system, the crosslinker will stay in the system. If you have them loose, they will kind of diffuse out from the hydrogel by time, and in that sense you have less crosslinkers, and then the nutrient stability will be quite low. If you have covalently bonded crosslinker, then it will stay in the system and you will have a more stable hydrogel. So that was our idea here. And in this case we use alginate instead of hyaluronic acid, and the reason for that was just that it's cheaper. But as I said in one of the lectures, it took quite some time for us to functionalize this the alginate. But we succeeded, so the work is done by the alginate here. So we functionalized it, we added aldehyde groups. We also actually in this system added arginine, which also can be attached to the hydrogel and through the same links. I won't show you any results of that, I think, but just to see how a drug can be released and diffused through this system as well. And what we could see that we were having the right ideas. I will tell you what this says. If you have the crosslinkers that is not attached to the alginate, then the stability is very low. The one here in the bottom where you have the red and blue, that is the ADH and PDO where you just have the crosslinkers added. So the stability there is quite low. If you have the one that is attached to the hydrogel on one side, then you have a higher stability. And also if you look into the mechanical properties, you can see that it will vary dependent on if you have the loose crosslinkers or if you have the crosslinkers that is attached to the hydrogel. So again, if just looking into the free crosslinkers, we can see that the mechanical properties is higher if you have the PDO. Now I get a bit afraid that we start to make a add sound here. Thank you. Thank you very much. So if you have the PDO, where you have the oxide again, as before, we can see that we have higher mechanical properties compared to if you have the ADH. We could also see that in the case when we have the crosslinker attached to the alginate, you can also vary the mechanical properties by just choosing different concentrations. The green and the violet is just different concentration of the polymers. So in that sense, we could also vary the mechanical properties. In below, we have a stress relaxation, and we can see that we can vary the stress relaxation if you have a free crosslinker. You have a much faster, a much lower stress relaxation compared to if you have the crosslinker attached to one side. Then you have a much higher stress relaxation. And looking more in detail into this, it's a bit interesting because if you have compared the red and green, they have quite similar mechanical properties, but in below you can see that they have very different stress relaxation rates. And this was interesting for us to see that you can actually tune individually the mechanical properties and the stress relaxation here. So the mechanical properties is not directly related to the stress relaxation rate. That was something that we really liked in this case. So then we have a system where we can play around with the mechanical properties. We can also make the hydrogel, it has a longer stability. So in the end there, our next step in this case is to, since we have the possibility to add coumarin entities, we try to use that in this hydrogels as well. So we have this, as I just said, we have in this case we're working again with hyaluronic acid. Then we functionalize it and we attach this ADH crosslinker. And then we have another macromolecule where we just have the functionalized hyaluronic acid with aldehyde. And then as a third component, we have this aldehyde functionalized, that we have the ADH crosslinker, but we have also this coumarin cage to attach to the hyaluronic acid. And what this means is that on the top, when we add these macromolecules, you have a hydrazide that becomes a hydrazone. And if you, here in below, you have the hydroxyamine, and when you irradiate, you create an oxide. If it reacts, if this one, the hydroxyamine that is created, if that one reacts with an aldehyde, it becomes an oxide. So just to take it again, if we just add these three components and we don't irradiate, what you then have in your system is an equilibrium between the hydrazide and the hydrazone, which is quite okay. It's what's the one in the middle that likes to be on both sides. If you then irradiate, you have these three components and you irradiate the specific points, then you create this hydroxyamine that will react with some aldehyde and form oxides in your system. And the oxides are more stable. So at those points where you create this oxide, we were thinking that these specific points you will have higher mechanical properties. So in this case, not to use this system, in this case we're not releasing growth factors, but we're actually deciding where in the system we want to have better mechanical properties. That is at least our ideas. It's not verified 100% yet. I will show you some results. You still need to look into and also how to characterize the mechanical properties exactly on these spots where we have irradiated. But you follow the system. And then, of course, depending on how much of these three different macromolecules we add, we can also vary the properties. But then when we started to evaluate this, we could see that the mechanical properties, when we irradiate it, so here it's not irradiated, and then we irradiate, the mechanical properties increases of the bulk. So you have a gel, and then you irradiate, and then we can immediately see that the properties increases. So we could verify quite easily that something is happening in the gel when we irradiate. We could also see that if we're adding more of the coumarin-based macromolecule, we also increases the mechanical properties. So it's nothing else that is happening in the hydrogel. It is something related to the irradiation and the amount of coumarin, which also made us think that it is this reaction that we were thinking in our DS that is actually happening. And we can also see that the stress relaxation time is longer after irradiation. So until now we're quite happy, and we can also see we have started to look into in the confocal and the irradiation using NIR that at specific points, since the coumarin base also is fluorescent, so we can also see that we have chosen different regions of interest, and where we irradiate, we can also see that the fluorescence is less. So we can say that we have a spatial thermal temporal control. We create these region-specific changes, and also that we can create gradients in the properties related to the rheology that I just showed you. So what we have to do now is to look into these region of interest to see if we actually get different mechanical properties there or not. If we succeed, I think that we have a really nice system to play around with. And in that sense, we have both possibilities to attach and release the growth factors using light and hydrolysis, and we can also play around with the mechanical properties in the hydrogels. For these ones, we have until now just looked into the surface, but since we could irradiate at different levels in the Z direction using, it's kind of the same system. I'm quite sure that we can do it here as well, so that we also in the Z direction can play around with the mechanical properties. So in that way, we are playing around with the environment in our hydrogels, and something that I want you also to inspire you to not only look into the surface and the mechanical properties of the scaffold, but also look into how to interact and optimize that in a better way. So that is actually what I plan to tell you, a brief introduction to that area, I would say. Questions or comments related to this?
How do you measure the self-healing efficiency? And you also said like we can use different dynamic covalent bonds other than oxime, hydroxide, and imines.
Yes.
Are they having cytocompatibility or any toxicity issue with other dynamic covalent bonds?
We have only tried this one because it's a good cell and material interaction. Also, that it's easy to functionalize proteins and growth factors using aldehydes. But we are starting now another project where we will evaluate other dynamic covalent bonds as well. Not only specifically towards tissue regeneration, it will be more broadened to see how to print different systems.
Yeah, with this one we can prepare 4D printable materials also.
Yeah, exactly. So yeah, for now we only have worked with the tissue phases, but in a year we will have a broader platform to play around with.
Actually, I was working on it.
Yeah.
I used the coronic ester linkage, metallic and coordination bond transfer reaction.
Yeah.
Can we? I don't know about the cytocompatibility. Drug release or something. I don't know more about the biology. But if that interaction is having compatibility, then we can explore in that direction also.
Yeah.
I wanted to know.
Yeah, I'm sure. I don't. I mean, I'm not a specialist yet on the other systems. We are really digging into that now, together with a postdoc, to see the advantages and disadvantages of the different systems that can be used. But yeah. So my answer to you is I'm sure that there are other good systems to use. Perhaps yours as well, but that I cannot.
Actually, I'm working on the tandem covalent bonds, but I don't know about the biology part. I'm more into material.
Yeah.
Like I also developed a prosthetic using the tandem covalent bond that can be like, which is recyclable. Like that was just a part of the study.
Yeah.
And I also prepared some other materials, recyclable and self-healing materials using this metallic and transmitter reaction. I was thinking like these interactions can be used to prepare 4D printer materials. Your next aim is that.
Yeah, sure. But you also asked about the self-healing, how to evaluate it.
Yeah,
But you do
how
that
much
as well.
efficiency, like here we used tensile strength of the twisting sample
Okay.
with self-healing sample. Like we are comparing its ratio. After self-healing, how much the tensile strength, like how much tensile strength it has. By like just a ratio, by we are taking ratio, comparing with the original work, we are measuring its efficiency.
Yeah. Yeah. I think it's
And we
good.
can also use the using the geometers.
Yeah. Yeah, that is what we did. But I cannot say that our method is better than yours. I guess perhaps it's also, yeah. Because we just see that it's healed by looking, visualize. We look at it and just make sure that it's healed, and then we do the rheology. Or that is what we have done until now. We didn't do any tensile tests. But
Any other questions?
I don't know more about chemistry and I do want to functionalize my hydrogel for the better release. So what could be the better way to start?
how to functionalize it. And you're using Germa. I don't know. I mean, an easy way is to just to add aldehydes, I would say, in some way to your gel. I mean, that is.
It's more due to the it's more related to the release of the protein. So how it will affect it? It is a bioactive molecule. I want to release it in a controlled manner.
And you want to attach it covalently?
Yeah. No.
Or
I'm printing it with inside the ink.
now I don't follow.
The protein molecule itself is in the bioink.
Yeah.
So I'm just 3D printing it.
Yeah.
So it is inside the ink. Now if I want to functionalize the scaffold for its better release, its controlled release.
And what do you mean with better release? Should it be slower? Or
Yeah, it should be slower. It should be at the defect site for the more duration, and then it should release slowly.
I guess there are many different ways, but if we just relate to what I presented here, what we did we had this arginine that could react with the aldehydes on the scaffold. So in that way we could make the release slower, because it doesn't just diffuse out, it also reacts with the scaffold, and then it will take longer time. There is something, but then depending on the equilibrium between the drug and the hydrogel, then you can decide how fast the release will be. So if you have a drug where you can have a function group that reacts with the aldehyde, and there is a covalently dynamic bond there, you can decide the rate. Yeah. You can also, of course, use covalent bond and use something that, as we did with the protein, that can be hydrolyzed. It's also an option.
Hope there are no more questions from the participants. And this is the last lecture of Professor Anna in this particular CAN course. requested her to be present in the valedictory function because I wish that you should get certificate at her own hands. So, and I requested Ingemar also to be present on this event. So she will be here at 4 o'clock during her valedictory function. And really we enjoyed all the rather lectures of Professor Anna. I told her already, but then, you know, when I was discussing with my colleagues in the morning, Ajinkya Bakshi is my colleague actually. He told me a very nice thing. He said, sir, even looking at the slide itself is a rather upgradation of all our knowledge, even how to prepare in slides and all that. And yes, that's true, because in this particular lecture, I'm not from chemistry, but I enjoyed the way you have actually shown everything nicely and very easy to understand. Just to know from your side, like as you said that we'll have to interact with the biologists, chemistry, chemist people, and then maybe the engineering side and all together. It is true in multidisciplinary, and I hope more and more discussion, more and more maybe the interaction always helps. So what I particularly feel that having a different departments are okay, but the faculty room should be in one block where all of them are sitting together. So that I do want the next cabin is not from the mechanical engineering or from the other engineering side, but maybe from the chemistry person, polymer person and all that. I think that actually creates a very good ecosystem for interaction. I think in every institute, if at all it is like that, it will be quite interesting. Otherwise, you know, when two of us sit together of the same department, maybe we don't talk about academics, we talk about some administrative matter and all that. And that actually doesn't upgrade ourselves. So we must appreciate Anna's efforts. So please join me.
Thank you.
So we'll have a break for some 10-15 minutes and then I will start my lecture. Till that time.